Thank you for inviting me to speak today on the topic of preparing for clinical trials in CMT. Over the past several years, there have been exciting advances in publications of potential treatment approaches for CMT across different forms of CMT and including by way of gene and other therapies. With these advances, it has really focused our attention on how we conduct trials in humans with CMT in the hopes of achieving multiple effective therapies. By way of background, clinical trials in humans are conducted in several phases. In phase 1 studies, we typically test potential treatments in a small number of individuals with a focus on determining the appropriate dosage and also beginning to assess safety. In phase 2 studies, treatments are tested in a larger number of individuals. The focus here is safety, but we also begin to obtain some information about the potential effectiveness of a treatment. However, before treatment can generally be approved by regulatory agencies such as the FDA, we as clinicians have to conduct phase 3 trials. These are typically conducted in a larger number of individuals. Usually there is a control group that receives a placebo. And the main focus here is confirming safety, but importantly, determining whether the treatment is effective or not, so-called efficacy. After successful phase 3 trials, therapies may be approved by a regulatory agency, but even after approval, there's continued monitoring regarding their safety and effectiveness through additional studies known as phase 4 studies. Our initial efforts to develop treatments in CMT focused on CMT-1A. A number of years ago, we conducted treatment trials of high-dose ascorbic acid, so-called vitamin C, in CMT-1A. We did this because of an animal model that suggested it might be effective. And this led to multiple trials of vitamin C for CMT-1A across the globe. In North America, we conducted a randomized double-blind placebo-controlled trial of vitamin C for CMT-1A. However, the North American trial, along with virtually all other studies of vitamin C in CMT-1A, were negative. We found that it was not an effective treatment for CMT-1A. But despite the negative results of these trials, we learned much about our limitations at the time in conducting clinical trials in CMT-1A. Firstly, we learned that CMT-1A progresses at a very slow rate, and thus it's difficult to detect change within a year or two in a clinical trial focusing on CMT-1A. We also learned that our various outcome assessments that we used for the CMT-1A vitamin C studies, and most importantly, the CMT neuropathy score, while useful to categorize different levels of severity of CMT, were not sensitive to change over time in CMT-1A. And therefore, we learned that we needed new, more sensitive ways to measure treatment effects in CMT in general and in CMT-1A in particular. Following the vitamin C studies, we formed the Inherited Neuropathy Consortium. This has been led by Dr. Michael Shai. We at the University of Rochester and other centers across the United States, Europe, the UK, and Australia have been actively involved in this. There are over 20 centers across the globe today. And the Inherited Neuropathies Consortium is focused on natural history studies and studies to develop new ways to measure treatment effects for various forms of CMT. The goal, of course, is to develop effective treatments. But first, we have to be able to conduct treatment trials in an efficient manner. In order to approve a treatment for any condition, and including CMT, the FDA indicates that the potential treatment should impact how an individual feels, functions, or survives. Fortunately, in most instances, survival is normal or near normal in individuals affected by CMT. And therefore, the emphasis of treatment is to improve how individuals function and how they feel. And we have set out in the Inherited Neuropathies Consortium, both for children and adults, to develop ways to measure how individuals with CMT function and how they feel, in the hopes that we can use these assessments in clinical trials to prove whether a treatment is working or not. So the Inherited Neuropathies Consortium, in work led out of Australia by Joshua Burns, has developed an assessment known as the CMT Pediatric Scale. This assesses all aspects of physical functioning in children with CMT and has 11 items shown here. Similarly, we have needed to develop a scale for infants. While most forms of CMT are onset symptomatically at an older age, some severe forms of CMT are onset at birth or in the first year of life. And the CMT pediatric scale is only appropriate for individuals 3 to 18 years of age. And therefore, again, in work led by the Inherited Neuropathies Consortium and Joshua Burns and Melissa Mandarakis and others, we've developed a scale known as the CMT Infant Scale with scoring that is very similar to the CMT pediatric scale in approach. However, we were lacking a similar scale and approach for adults. And thus, based on our CMT pediatric scale, we've developed something in work led out of the University of Rochester known as the CMT Functional Outcome Measure, or CMT-FOM. This includes several items of upper limb function shown here, including grip strength, fine motor function through a 9-hole peg test and a functional dexterity test shown here. We also include multiple measures of gait, balance, and mobility shown here. This includes quantitatively assessing ankle strength, testing balance in different ways, and testing mobility in using different approaches. We hope that the CMT functional outcome measure, along with the CMT pediatric scale and the CMT infant scale, will be the primary way that we assess whether treatments in clinical trials are positively impacting function. But also important beyond what we can measure in terms of an individual's function is the individual's perspective on how CMT is affecting them. In other words, the individual's voice in the assessment of potential treatments in clinical trials. We do have a patient quality of life scale for children with CMT and a caregiver version of that, but we were lacking a scale for adults. And so again, in work developed through the Inherited Neuropathies Consortium and led out of University of Rochester, we've developed a questionnaire known as the CMT Health Index. This was developed according to FDA guidance on how to develop questionnaires to assess quality of life. And it was developed after extensive input from individuals affected by CMT. Not surprisingly, individuals with CMT rate problems with foot and ankle weakness, mobility limitations, inability to do activities, and problems with hand and finger weakness, as well as impaired balance, as the most impactful and challenging issues that individuals with CMT face. However, in developing the CMT Health Index, we learned that individuals with CMT face a number of challenges across different aspects of day-to-day life. What I've discussed up to now have been what we term clinical outcome assessments, assessments of how individuals feel and how they function. But another form or approach to assessing whether treatments are working are known as biomarkers. Many of you may have heard the term biomarkers. So what are biomarkers? Well, a biomarker is a biological marker in blood, other body fluids or tissue that indicates the state of a condition. And biomarkers can be used to see how well the body is responding to a treatment. And importantly, we're interested in biomarkers because they may provide a very early indicator of whether the treatment is working. For example, a biomarker measured in blood or some other bodily fluid may begin to change in response to a treatment before an individual notices that their symptoms are improving or before a physician can identify improvements on the clinical examination. There are many different types of biomarkers, and we in the Inherited Neuropathies Consortium are studying these in many ways. There are biomarkers that we can measure in blood or plasma. One that you may have heard of is neurofilament light chain. This tells us about the state of nerve damage, and it is a promising biomarker for use in CMT. We can also measure biomarkers in biopsies. For example, there's much interest in looking at different proteins and mRNA levels in skin biopsies. For example, in CMT-1A, which is due to an extra copy of the PMP22 gene, we can measure expression levels of PMP22. Too much PMP22 is detrimental to individuals with CMT-1A. And in the future, if there are treatments, genetic or other, that lower PMP22 levels, we may be able to measure that lowering of the PMP22 early in the course of a treatment trial and pick that up before an individual notices that their symptoms are improving or before a physician can identify improvement on clinical examination. We've also been doing research on imaging biomarkers. We've identified in the Inherited Neuropathies Consortium and in work led out of the University College of London by Dr. Mary Riley and Dr. Michael Shai from University of Iowa, that MRI of leg muscles can be a very helpful way to determine progression in CMT-1A. And we hope that by imaging leg muscles with MRI, we may be able to detect treatment effects early in the course of a clinical trial. Similarly, individuals with CMT-1A not only have problems with muscle atrophy and weakness and foot drop, but they also have difficulty with sensation. And lack of sensation can lead to poor balance and difficulty with hand activities. We at the University of Rochester have developed a technique to actually visualize painlessly with a confocal microscope sensory nerve endings in the skin in humans, known as Meissner corpuscles. These are the sensory nerve endings in the skin of the hands and the feet that allow one to feel touch pressure. And therefore, we are studying this to determine whether potentially this can be used in a treatment trial to assess treatment effects. Finally, there are other forms of biomarkers known as electrical or digital biomarkers. Nerve conduction studies that many of you may have undergone in diagnosing CMT are one form of electrical biomarker that can be followed in a clinical trial. There are also other biomarkers known as wearable sensors. For example, a Fitbit or other kinds of sensors that can determine one's activity levels. All of these may have potential value for use in clinical trials to identify whether a treatment is working or not, and we're conducting studies across all these types of biomarkers. One area of focus of ours has been to accelerate clinical trials in CMT-1A through a study that is led by myself out of the University of Rochester, known as ACT-CMT. This is supported by the NIH and involves multiple centers within the Inherited Neuropathies Consortium within the United States, the UK, Italy, and Australia. The goal of ACT-CMT is to address gaps in clinical outcome assessments and biomarkers for CMT-1A trial readiness through multi-center validation of the CMT functional outcome measure that I described for you, The CMT functional outcome measure is shown here. A second aim is to confirm whether MRI of calf and thigh muscle fat content will be an effective way to measure progression of CMT-1A in clinical trials. And thirdly, we're assessing whether reflectance confocal microscopy of those sensory nerve endings in the skin known as myoclonic corpuscles, shown here in panels B and C, are a way to detect progression in CMT-1A and whether we might be able to use this in clinical trials. So shown on the right is an individual with multiple of these sensory nerve endings or myoclonic corpuscles in their skin. In the center panel, panel B, is an individual with mild CMT-1A who has lost some of their myoclonic corpuscles. And to the left, panel A is an individual who has no remaining myoclonic corpuscles, is an individual with severe CMT-1A. Additionally, I discussed wearable sensors in CMT. We are, through a study at the University of Rochester supported by the CMT Association, assessing whether a novel wearable sensor known as the BioStamp MC10 endpoint system can sensitively assess aspects of gait and balance. My colleague, Dr. Kate Eichinger, is leading this and conducting studies both in clinic where individuals wear these sensors, and through computerized means, we're able to obtain very detailed quantitative information about walking and balance ability. We're also asking individuals to take these home with them, and in the home environment, we're able to obtain similar information about gait and balance function. Through other studies in the Inherited Neuropathies Consortium, we're evaluating different types of sensors. The actigraph shown here is a sensor that can be worn at the hip for a week and can be used to assess physical activity. We know that individuals with different forms of CMT have reduced physical activity compared to individuals who don't have CMT because of the disability of their related to their CMT and because of pain and other factors. And so with the actigraph, we can, in the home environment, assess step count, the time individuals are engaged in different forms of physical activity and the different intensity levels, and time spent sitting and sleeping, for example. Another form of sensor that we are studying is the so-called APDM, gait and balance system. This is shown here. The sensors are worn on the chest, waist, arms, and feet. And in the clinic, we can do a very detailed analysis of walking and balance function. And we can obtain information about, for example, stride length, step speed, and the amount of sway with balance activities. By using all of these sensors, we hope to obtain detailed quantitative information that we hope will provide information on early changes in gait and balance, hopefully in the setting of treatment trials. We, of course, cannot conduct treatment trials beyond a couple of years in individuals. This is quite burdensome. And so the hope is, with all these measures I've discussed, including some of the functional outcome measures, the patient-reported outcome measures, the CMT Health Index and others, and with the biomarkers and the wearable sensors, that we will find methods to very rapidly determine whether treatments are effective and whether they should be then carried forward to larger phase 3 trials. Now, I've spoken mostly about work that's being done through the Inherited Neuropathies Consortium and the ACT-CMT study in CMT-1A. But there is extensive work ongoing at our research centers in multiple other forms of CMT, including CMT-1B, CMT-2A, CMT-X, and multiple rarer subtypes of CMT. All of this is being done so that we will be ready to conduct effective trials in different forms of CMT, as hopefully in the coming months and years, there are multiple treatment candidates that will be ready for testing. You may ask how you can participate in CMT research. Hopefully, many of you are already participating in CMT research. But if you've not had the opportunity, you can participate through the Inherited Neuropathies Consortium at the URL shown here. Also, if you're interested in participating in our studies at the University of Rochester, Beth Wood can be contacted via email or telephone. And thank you for allowing me to speak to you today on preparing for clinical trials in CMT.